Hey everyone, this is my Lesson 8 group problem video. Just to go over the problems here. We have our first problem says that we are identifying the allele composition of the four products of meiosis. So if we take the normal chromosome that they give us here, and then we take the inversion chromosome. This is the diagram that it's going to look like here on my homework that I drew out. And so after crossing over, we'll get one allele composition of the normal chromosome, the capital LMN, OPQ, RSTU, VWX. And then our second allele composition for our second chromosome is going to be the same starting point of the chromosome, the capital LMN, and then it's going to cross over and it's going to actually contain this first part of the inversion chromosome, the lowercase LMN. And then for the third chromosome composition, kind of the same thing, except we're going to take the last part of this chromosome here, the capital XWVUTSRQPO, and then it's going to cross over with the last part of the inversion chromosome, the lowercase OPQVUTSRWX. And then our fourth chromosome allele composition is just going to be the same composition as the inversion chromosome, the lowercase LMN OPQVUTSRWX. And for problem 2, for fill in the blanks, a non-disjunction in meiosis 2 produces a 50% aneuploid gametes, and we can find that in the notes here when we are talking about the, let's see, when we're talking about the non-disjunctions here in, basically it produces 25% monosomy chromosomes and then 25% trisomy chromosomes. So it's going to produce a 50% aneuploid gametes, whereas in mitosis, that produces a mosaic phenotype. And for B, meiosis is an individual who has suffered a paracentric inversion because it produces a dicentric chromosome and also an acentric chromosome. And we can find that in the notes right here. So yeah, it creates a individual with a dicentric and acentric chromosome. And then for C, I have the Turner syndrome is an example of a human disease caused by monosomy, while the familial Down syndrome is an example of a disease caused by a Robertsonian translocation. And a familial Down syndrome is different than the trisomy 21 Down syndrome because it has that Robertsonian translocation. And for D, we learned that the human liver cells can be the 4N, 8N, or 16N. And for E, a terminal deletion in chromosome 5 causes the Cre-du-Chat syndrome, while an inversion on the X chromosome causes that type A hemophilia. And for F, I have that a duplication is a change in chromosome structure that can be potentially beneficial to an organism or an entire species, because as we reproduce and have more chromosome options when we duplicate our chromosomes, then we have more likely chance to create a like survival gene. So duplication is can oftentimes be beneficial to organisms. And then for G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offspring. And we can actually see this in the notes here. The notes, where are they? There they are. Segregation. Yeah, the alternate segregation, after meiosis 2, there are 2 normal gametes produced that all of these can produce the viable offspring. All right, and then for question 3, we have that in Turner syndrome, that's going to produce 45 chromosomes. And in Jacob syndrome, it's going to have an extra chromosome for one of the chromosomes, so it's going to be 47 chromosomes. And the Cre-du-Chat syndrome does not affect the number of chromosomes in an individual, so it's going to stay at that 46 chromosomes. And familial Down syndrome, that's when it has the one less chromosome, like one of them doesn't duplicate, so we're going to have 45 chromosomes there. And then type A hemophilia also does not affect the amount of chromosomes in an individual, so that's going to stay at 46 chromosomes. And for B, in the Charcot-Marie tooth disease, type 1A, I had that the terms not associated with this are going to be the triploid, reciprocal translocation, and non-disjunction. And then for C, the Klinefelter syndrome, I have that the terms not associated with that are going to be triploid, chirocentric inversion, reciprocal translocation, and non-disjunction. And then for D, the terms that are non-associated with type A hemophilia, it's actually going to be all of the terms here because type A hemophilia is not affected by like chromosome number. So none of these terms are going to be associated to type A hemophilia. So I just put all of those there. And for 4, so they're asking for the diagram and the synopsis of these chromosomes here in a reciprocal translocation. So in a reciprocal translocation, I have the diagram here where we have a normal chromosome, which I put as number 1. So that's just the homologous chromosome 1, and then the homologous chromosome 2 is going to be this one right here, and that's going to be the blue one. And then when a reciprocal translocation occurs, we're going to come out with chromosome 3 here that they give us in the problem, and then chromosome 4, and I have diagrammed these as chromosome 3 here, and then chromosome 4 here. And you can see how these contain part of chromosome 2 and then also a little piece of chromosome 1. And then for this one, it contains chromosome 1 and then just a little bit of chromosome 2 right here. So that's what happens in a reciprocal translocation after in the meiosis process. And for B, it says to identify the allele composition. So in the alternate segregation, the allele composition is going to be chromosome 4 and chromosome 3, they're going to be in the same daughter cell. So that's going to be the MNOGH centromere IJKL, and then going to be the DEP or DEFP, and then centromere QRST. So that will be in one daughter cell, and then the other daughter cell is going to have chromosome 1 and 2 in it. So it's going to have that DEFGH centromere IJKL, and then the MNOP centromere QRST. And then for the adjacent 1 segregation, the allele composition for that, we're going to have one daughter cell with the chromosome 4, which is the DEFP centromere QRST, with chromosome 1, DEFGH chromosome centromere IJKL. And then in the other daughter chromosome here, it's going to have chromosome 3 and chromosome 2. So that's going to be the MNOGH centromere IJKL, and then MNOP centromere QRST. And then in the adjacent 2 segregation, the gametes, we're going to have one daughter cell with chromosome 4 and 2 in it, and then the other daughter chromosome or other daughter cell is going to have chromosome 3 and chromosome 1 inside of it. And for number 5, we're identifying the total number of chromosomes in a cat. So if a cat is triploid, that means that it's 3N. So N being 38, 38 times 3 is going to be 114 chromosomes. And a cat that is monosomic, for chromosome 12, we're just going to have one copy of chromosome 12. So it's going to be one less of the original 38. So we're going to have 37 chromosomes. And for a terminal deletion on chromosome 7, we're not completely getting rid of the chromosome itself. It's just a part of that chromosome. So we can still account for that chromosome. So we're not going to have a difference in chromosomes. It's going to stay at that 38 chromosomes. And a cat that has suffered a Robertsonian translocation between chromosome 17 and 18, we're going to have one less chromosome in this translocation, so we're going to have that 37 chromosomes there. And then a cat that has suffered a reciprocal translocation between chromosome 6 and 9. In reciprocal translocations, just like in problem 4 here, we're not getting rid of any chromosomes, so it's not going to affect the number of chromosomes, so it's going to stay at that 38 chromosomes. All right, well, that is lesson 8. Thanks for listening.